My dear viewers, welcome to Biology at Priyojit. In today's class, we explore how the body manages proteins through three key processes: transamination, deamination, and the urea cycle. You will learn how amino groups are transferred between molecules, how excess ammonia is safely removed, and how the urea cycle protects our body from toxicity. By the end, you will clearly understand how these steps maintain nitrogen balance and support healthy metabolism. Amino acid oxidation. Amino acids are important fuel molecules contributing significantly to metabolic energy through oxidative degradation. First step is the energy contribution varies by organism. Carnivore up to 90% of energy from amino acids after a protein-rich meal. Herbivores they use amino acids for only a small fraction of energy. Microorganisms they can use amino acids as fuel when they needed. Plants they rarely oxidize amino acids for energy and regulate amino acids mainly for biosynthesis. In animals, amino acid degradation occurs in three situations. First of all is the normal protein turnover. What happens? Excess released amino acids are oxidized. High protein diet, surplus amino acids which cannot be stored are catabolized. And starvation and diabetes, when carbohydrates are unavailable, body proteins are broken down for energy. Common initial step: amino acids lost their amino group, forming alpha keto acids or carbon skeletons. Fate of carbon skeletons, that is alpha keto acids, it is oxidized to carbon dioxide and H2O, or it can be converted into 3 or 4 carbon intermediates such as which are needed for gluconeogenesis and glucose production. General features of amino acid catabolism: the pathways are very similar in most organisms. In vertebrates, best studied carbon skeletons enter central metabolic pathways, especially the citric acid cycle. And some degradation steps resemble fatty acid catabolic reactions. This is the image showing how the intracellular protein is converted into amino acids and vice versa. The dietary protein is also converted into amino acids. Then amino acid will be break down and it will form amino group and the carbon skeleton. The amino group will be utilized in the biosynthesis of amino acids, nucleotides and biological amines. And it will produce carbamoyl phosphate. The carbamoyl phosphate then enter into the urea cycle. During the running through the urea cycle, a nitrogen excretion product urea will be formed. And then the urea cycle is also linked with the citric acid cycle through aspartate argininosuccinate shunt of the citric acid cycle. So again the carbon skeleton of the amino acids will be converted into alpha keto acid and it will enter into the citric acid cycle. During the running through the citric acid cycle, it will produce carbon dioxide, H2 and ATP. At the after the citric acid cycle, it will also oxaloacetate and which will be ultimately converted into glucose by means of gluconeogenesis. So what is the metabolic fates of amino groups of the amino acids? Nitrogen is very abundant in the air but too stable to be used directly by most living organisms. Only some microbes can turn nitrogen into unusable forms like ammonia. Amino groups in living systems. Amino groups come mostly from dietary protein. They are carefully managed because nitrogen is precious for the body. Where metabolism happens, most amino acids are broken down in the liver. Ammonia released can be reused for making new molecules or for biosynthesis, and it excreted directly or converted to urea or uric acid depending on the organism. Transport from other tissues. Ammonia from other tissues just outside the liver is carried to the liver as amino group to be safely converted and excreted. Key molecules in nitrogen support: glutamate and glutamine are the main carriers of amino groups. In liver cells, amino groups from other amino acids are transferred to alpha ketoglutarate and which will produce glutamate. The glutamate then releases ammonium ion or NH4 plus within mitochondria. In other tissues, ammonia is added to glutamine which moves to the liver for processing. Special role for muscles. The excess amino groups in muscles are transferred to pyruvate. It will produce alanine. Then alanine carries nitrogen to the liver and it is also called the alanine cycle. Proteins will be converted into amino acids. Then the amino groups of the amino acids will be transported to liver and it will be excreted as urea or ammonia or uric acid. Pyridoxal phosphate and amino group transfer. First step in amino acid breakdown in liver. Removal of alpha amino groups from amino acids by enzymes called aminotransferases. The enzyme is named as transaminases. Amino group is transferred to alpha ketoglutarate and it will form L-glutamate. Remaining part of the amino acid will produce alpha keto acid. So what is the purpose of transamination? It collects amino group from many amino acids as glutamate. Glutamate can then donate its amino group for biosynthesis and send amino groups to excretion pathways such as urea, etc. Then aminotransferases, it is specific for the amino acid donor and often for alpha ketoglutarate as acceptor. The reactions are reversible, which is equilibrium constant is nearly 1. So examples include alanine aminotransferase or ALT and aspartate aminotransferase as AST. Coenzyme, the pyridoxal phosphate (PLP) or vitamin B6 is essential. It acts as intermediate carrier of the amino group. There are two forms of the pyridoxal phosphate or PLP and one is the aldehyde form. It accepts amino group and the pyridoxamine phosphate or PMP donates amino group to the alpha keto acid. Covalently it is attached to enzyme via Schiff base with the lysine residue. Chemical role of PLP, it stabilizes highly reactive intermediates or carbanions during reactions at alpha, beta, gamma carbons of the amino acids. It supports reactions like transamination, decarboxylation and racemization, that is the conversion between L form and D form of amino acids. So what is the ping pong mechanism of aminotransferases? Amino acid binds, it donates amino group to pyridoxal phosphate and leaves as alpha keto acid, and then alpha keto acids will accept amino group from the pyridoxal monophosphate and it leaves as amino acids. So nitrogen excretion and the urea cycle we will discuss now. Forms of the nitrogen excretion. Different animals excrete nitrogen in different forms. Such as ammonotelic animals, they excrete nitrogen as ammonia. Examples: most aquatic animals, bony fishes, etc. Ammonia is toxic but easily diluted in water. Ureotelic animals, they excrete nitrogen as urea. Examples: humans, other mammals, most terrestrial animals release or excrete nitrogen as urea. They require less water and they are less toxic. Uricotelic animals, they excrete nitrogen as uric acid. Examples include birds, reptiles, etc. They saves maximum water. Plants, they recycle almost all amino groups and excretion occurs rarely. So what is the overview of the urea cycle? It occurs only in the liver. It converts toxic ammonia into urea. Urea travels through blood, moves into kidneys and then excreted in urine. So the cycle discovered by Hans Krebs and Krebs-Henseleit in 1932. Where the urea cycle occurs, the step 1 occurs inside mitochondria and step 2 to 4 occurs in the cytosol, and then cycle therefore spans two different compartments, cytosol and mitochondria. Steps of the urea cycle. There are 5 enzymatic steps. The first step is the formation of the carbamoyl phosphate. This is the carbamoyl phosphate. Inside, this occurs inside mitochondria. NH4 reacts with carbon dioxide and 2 ATP with the help of the enzyme carbamoyl phosphate synthetase. It is regulated step of the cycle. Then from carbamoyl phosphate, citrulline will be formed with the help of the enzyme ornithine transcarbamylase. Carbamyl phosphate will react with ornithine and it will form the citrulline. This is the citrulline compound, and then citrulline moves from mitochondria into the cytosol. Argininosuccinate formation, it is the third step. So the argininosuccinate synthetase enzyme reacts on citrulline and adds aspartate and ATP with it to form the argininosuccinate. Then argininosuccinate will be cleaved with the help of the enzyme argininosuccinase and it will form arginine and fumarate, and it is the only reversible step of the cycle. Then a fumarate will again enter into the Krebs cycle and the arginine will form the urea with the help of the enzyme arginase. Arginase acts on arginine to form urea and ornithine. Then ornithine returns to again mitochondria and it will start the urea cycle again. So what this is a metabolic channeling. Enzymes are arranged close together in the urea cycle in the different intermediates such as citrulline, argininosuccinate, arginine, ornithine pass directly between enzymes. Prevents dilution in the cytosol and it increases efficiency. There is a link between urea cycle and citric acid cycle. Fumarate, that is form of urea cycle, is converted to malate and it will form the oxaloacetate. Then oxaloacetate will be converted into aspartate and vice versa via the process transamination. And then aspartate fits back into the urea cycle. The link is the aspartate argininosuccinate shunt. Regulation of the urea cycle, long term regulation. High protein diet increases urea cycle enzymes, and starvation causes the breakdown of muscle and increases urea production. Low protein diet decreases enzyme levels. Short term regulation, CPS1 is the enzyme which is activated by N-acetyl glutamate or NAG, and NAG synthesis increases when glutamate and acetyl-CoA is increased, and arginine will be increased as an activator. So what is the energetics of the urea cycle? Energy cost total 4 high energy phosphate bonds per urea will produce 2 ATP for carbamyl phosphate and 1 ATP for the conversion of AMP to PPI for argininosuccinate formation. Then in how energy compensation occurs, fumarate will be converted into malate and subsequently it will converted into oxaloacetate and produce 1 NADH. We know that 1 NADH is equivalent to 2.5 molecules of ATP, and this reduces the net energy cost. Genetic disorders of the urea cycle. We know that defects prevent conversion of ammonia to urea, which causes the situation named as hyperammonia. Identify disorder by measuring which intermediate accumulates, because most steps are irreversible. Common treatment, sodium benzoate can be used. It combines with glycine and produces hipurate and it is excreted. It uses ammonia to regulate to regenerate glycine. Then phenyl butyrate or phenyl acetate, they combine with glutamine and form the phenyl acetyl glutamine, which can be excreted and it consumes ammonia to regenerate glutamine. Then carbamyl glutamate, it is used when N-acetyl glutamate synthase is deficient and it activates CPS1. Arginine supplementation, it helps in deficiencies of ornithine transcarbamylase, argininosuccinate synthetase, and argininosuccinase. Arginase deficiency, arginine must be limited in diet. This is the image showing the fate of amino acid carbon skeleton. So to conclude or to summarize, protein metabolism, which involves 3 essential steps that help the body safely manage amino acids. Through transamination, amino groups are transferred to form useful compounds. Deamination removes amino groups and the urea cycle converts toxic ammonia into urea for excretion. Together, these processes maintain nitrogen balance and protect the body from toxicity. Hope this class clarified the concepts. Stay curious and keep exploring biology. Thank you.